So unfortunately, Burkhard cannot be here, but for a positive reason, he just became a father. So I start with telling you that this talk is not about the origin of proteins, no synthetic chemistry here. It's about protein functions. And also I will not deal with the question, sort of historical question about what was the first polymer and at which point RNA and proteins emerged. Instead, I want to ask a number of questions that are sort of interesting. We know that all contemporary proteins are fairly big, and on the other hand, in the origin of life, we expect small proteins. So the question, and if you try to truncate contemporary proteins, they lose their activity. So can catalytic proteins be small? Most theories of the emergence of proteins, no matter which side you start with, assume that at some point there were random polypeptides. Again, the question is, can they be a source of functionality? How can we get a diversity of protein functions that we need for a minimal life? Is everything discovered separately, or some functions can change to another functions, something that in contemporary proteins does happen, but happen very rarely. Finally, what makes an evolutionary good protein? So until very recently, that was a realm of speculations. What was a real breakthrough was a technique that was developed in Jack Szostak's lab to do protein evolution in a test tube. And this is essentially the Selex technique very similar to what is done with RNA, but with a twist. So here is a slide that is essentially another version of a slide that Jerry showed on Monday, also very simplified version of what is done with an RNA. So you start with a very big library of RNA molecules, do selection according to some criteria. You keep what passed your selection criteria. You amplify it, perhaps you amplify it with mutations. Then you go through another circle using perhaps more stringent selection criteria, and you go in the circle until you're happy with your results. Well, that's all nice, but proteins do not replicate, so obviously that's not going to work for proteins in exactly the same form. And the trick is that this very interesting observation. I don't know whether my pointer works. I think it died, but that's okay. So, in the normal translation, mRNA is read by a ribosome and produces a protein, and at the end, protein and mRNA go their separate ways. But if you put puromycin at the end of your gene, then of your mRNA, then mRNA never separates from the protein. The protein stays, and you get this kind of a construct of a protein with the mRNA that codes for this, and that opens a possibility of using the same cycle in a slightly different way, which is that you do this, start with a very large library, you do this protein synthesis, you inactivate possible catalytic activity of RNA by making a complementary strand. Then the selection is on the protein. You throw out the protein, you amplify RNA that codes for this protein, and perhaps mutate, and again you go in cycles when selection is on the protein, but nucleic acids are being amplified. When you're happy, you keep your protein, you separate it from RNA, you sequence it, you test its activity just to make sure that it does what you want it to do. So you can start with a completely random library, or you can start with a library that is based on some existing protein. And the first try was on the random library, and that was to select for ATP binding protein. And what was found was that there are, at the end, 4 different families of ATP binding proteins were found. The probability was about 1 to the 10 to the minus 11. If you want to kind of know whether it's a low or a lot, it's about the same chances as finding a ribozyme. And so what it tells us is that a random sequence polypeptides could be a source of functionality. But what is really interesting is that these 4 families were completely unrelated to each other, but also in terms of the sequence, they were unrelated to any known protein. One of those families was actually fully characterized. And here is eventually high-resolution structure, both crystal structure and NMR structure were found. And so you see this ATP here, and that's the protein. It's a good-looking protein, actually. So it's a compact, good-looking protein, what was found. But this protein, sort of, you can find this motif in some proteins, but it does never exist as a separate domain in any contemporary proteins. But the question is, okay, is it really a good protein? And this is just to, so is it evolvable? And of course, if it's not evolvable, it's not very good, because if you're a protein and you're not evolvable, sooner or later somebody will come who will do the same job better than you do, and you will get eliminated. And a simple evolution process would be to evolve it from ATP binder to GTP binder, and that didn't work. But that was before the crystal structure was known, and when high-resolution structure came up, it became clear that this protein binds ATP through this small loop, but it binds it through the backbone. So hydrogen bonding is through the backbone, and so of course you can change residues till you're blue in your face, nothing happens. But you can redesign it computationally. And you have to take, of course, into account the fact that if you go to GTP, GTP is more soluble in water, so you have to by 3 to 4 kilocalories per mole. So binding to the protein also has to be stronger, which you can do by either improving stacking or probably both, or adding a hydrogen bond the same way as it happens in GC versus AT base pairs. And to do that, you have to, in this computational redesign, you add one residue to the loop, then the loop can invert itself. Residues, side chains can interact with the purine ring, and then you can get this extra hydrogen bond. But it's not all you have to do. You have to do a few other things. You have to keep the structure together. You have to close, since you make the loop more loose, you have to close access of water. So there are several mutations that you have to include to make this work. Now, another selection process, and that's selection of RNA ligase from the previous protein required zinc. This is a double zinc finger, and you can do RNA ligation. That's again, ribozymes exist, and Jerry talked about them a lot. And this time you start with an existing motif and you mostly randomize this. You could do it for ATP binder and it works. You can select ATP binders this way, but can you select an enzyme, which is a much more difficult task? And the answer is yes, you can. And there is a lot of mutations that go on, and in particular, there are mutations in the sites cysteines, and cysteines coordinate zinc, so it tells you that something changed in the structure, important change in the structure. And here is the structure. And the structure is new, so such a fold does not exist for any known proteins, but it looks very different from any proteins we know, because there are two parts that are very ordered, and that's where zinc sits, and a very flexible part, a loop, that's why there is so many, it's an NMR structure. Many different structures are observed. So we can kind of quickly summarize what was here. So we found some things that are very different from, in terms of evolution of a protein, very different from what we know about modern protein evolution. One of them is that, at the example of ATP binding protein, we can get a single function from multiple structural and evolutionary solutions. Normally, in contemporary proteins, we have one function. Usually there is pretty much one structure, sometimes two or three structures, and this is actually the basis for genetic annotation of proteins when we do genomic analyses. So that's quite different. And it seems that there are many different ways to find different proteins. But also the opposite is true. The opposite is that you can start with, you can get multiple functions from a single evolutionary origin, and so you can get an ATP binder, or you can get a ligase starting pretty much from the same scaffold, and there are probably many, we tried just one scaffold, but there are probably many different scaffolds that will have a very similar properties.